In our next lesson on transcription and RNA, we want to look at the mechanism of RNA polymerase. Here we have on the upper right a cutaway schematic of the structure of RNA polymerase in its different parts. So let's look at how these parts function together in the process of transcription. As you can see, the DNA is pictured as in blue and green. Double-stranded DNA enters the active site and the strands separate. Remember, we're only reading one of the strands. The template strand pictured in blue makes a right-angled turn. There is a clamp pictured at the top of our figure here that closes snugly over the DNA template, and that's thought to contribute to the high processivity of the enzymes. In other words, it doesn't let go. It continues transcribing and moving from one nucleotide to the next. You can see the clamp a little bit more clearly in the figure on the lower left here in this ribbon diagram. The nucleotide triphosphate substrates enter RNA polymerase through a pore at the bottom of the screen here. Of course, those nucleotides will hydrogen bond with the templates, and that's how RNA polymerase knows that it's adding the correct nucleotide on the growing RNA chain. The correct ribonucleotide triphosphate causes a loop of the protein to close over it, and that's how we ensure the accuracy of transcription. In other words, this is how RNA polymerase determines it's added the correct nucleotide. The nucleophilic attack is identical to that of DNA polymerase in that the 3' OH on the nucleotide that was last added to the RNA chain attacks the 5' phosphate of the incoming nucleotide triphosphate. And of course, synthesis occurs in the 5' to 3' direction. In the process of forming the RNA molecule, there is a hybrid helix that forms. That is, one strand DNA, the other strand the newly synthesized RNA. As we look at the structure of this hybrid helix, is actually a mixture of A and B DNA, and that's illustrated at the lower right of the screen here. On the far right, we have B DNA. Remember, this is the most common form of our DNA. A DNA on the far left occurs under dehydrated conditions and has slightly different structure. And you can see the DNA-RNA hybrid is kind of a mixture of the two. Remember, for RNA, there's an extra oxygen atom at the 2' position, and that constrains the hybrid molecule so that it can't make the same tight contacts that it does when both strands are DNA. This means that the double helix of DNA and RNA is wider and flatter of DNA and DNA. This makes it less stable and it dissociates more readily. But this is actually desirable in the process of transcription because we're not interested in forming a stable permanent structure. We only need it for it to hybridize for the short time that it is contacting RNA polymerase. At some point, we want to separate the two strands so that the RNA can exit and leave RNA polymerase. How do we keep things moving in the process of transcription so that we move smoothly from one nucleotide to the next? There is a helix near the active site called the bridge. In the figure on the right here, you can see most of our structure is in gray. You can see that green alpha helix of the bridge here. It oscillates between a straight and bent conformation, and that's thought to ratchet the template so that we can add the next nucleotide, so that we don't skip a nucleotide. We move from nucleotide to nucleotide. Remember, if we skip a step or we add extra, we've altered the genetic code and therefore we've altered the protein product. How do we release that RNA product? That's illustrated on the left here. There's actually a rudder protein within the complex. It's a protein loop that helps to separate the RNA, pictured here in red, from the DNA so that it exits through the portal here and dissociates from RNA polymerase. What about that proofreading ability? As the RNA molecule is formed by RNA polymerase, That hybrid helix is distorted if the wrong nucleotide is added, and RNA polymerase can sense this. Synthesis then stops and RNA actually backs out of the active site. And that's in our illustration here. You can see the backtracking of the RNA in the dotted red line. Now, RNA polymerase does not have exonuclease activity. All it does is sense that the wrong nucleotide has been added, and it exits the RNA out of the active site. To remove the wrong nucleotide, we need the action of a transcription factor, TF2S. It has nuclease activity. It trims off the wrong nucleotide so that RNA polymerase can begin again to add the correct nucleotide. In our next video lesson, we'll see how transcription moves from initiation to elongation, and we'll also look at the mechanism for transcription termination.